chlorophyll from the line height and chlorophyll from extract on a log-log plot. It looks fantastic. It was actually still pretty good. I could validate by looking at the model estimated phytoplankton absorption versus the one that I measured on the filter pad. Again, over a large dynamic range, looked pretty good. And backscattering, we didn't have backscattering sensors then, but I had a Coulter counter. And so I actually used the Coulter counter and calculated the particle cross-section and compared it to backscattering. I should have gone further and actually done the full me code, but I didn't know how to do that back then. But that actually, the relationship between the particle cross-section and backscattering was probably the best in terms of validation. And that's on a linear scale, right? So that was pretty encouraging. All right, so then the question is, so I can do pretty well estimating the IOPs, but what about the spectral variations? Because this was what the assumption of the curve was. This was some constant shape. And so what I did is I went and I looked at the difference between the modeled and the measured, and I converted it back to an absorption equivalent and added it back into the model result. So again, you know, like where they're different there, the measured was higher, the model over estimated absorption, so I removed that amount of absorption from the model spectrum. And so here are the same 4 examples. So the thin curve is measured on a filter pad, and the black curve is the model, which was this shape, the green shape, minus the residual. And so you see that it actually allows you to get much more peaked here. There's like that little feature there of absorption. It's starting to give you a little bit of that type. This was more flat. This one was quite peaked in the measured, and it kind of overestimated the peaked. But you can see that it allows you to get some of this extra information that Ken was talking about from irradiance. And so, you know, wherever your model doesn't fit, that becomes an opportunity, right? When Emmanuel was talking yesterday about signal and noise, and someone's signal is someone else's noise, well, you know, look at your residuals. There's often a lot of information in your residuals. Okay, and then I did a sensitivity analysis, the best that I knew how to do at the time, and basically I just changed the shape of the spectra, reran the model, and tried to figure out what my uncertainty was. And basically I still could get within 30% coefficient of variation. And phytoplankton was the most robust thing that I could retrieve based upon the measurements. And why do you think that that is? Why do these models all tend to do phytoplankton really well and tend to not do CDOM, NAP, and backscattering as well? Do you have an idea? Yeah. Yes. Yep, exactly. And you get a lot of transference of variance mathematically if your equation is the quotient, right? So you could have a little bit more or less of this balanced with a little bit more or less of this, and where does it show up? I mean, the model, the regression doesn't know. It's just trying to fit a minimization. And so where it really fits is where you have phytoplankton with lots of shoulders. And the Levenberg-Marquardt, which is the method that I used, actually does a fit on the derivative. So it's really tuned to trying to identify where those shoulders are. And so if that's what you're after, it's a really excellent approach to least squares regression. So, okay. So the big assumptions are the spectral shape of your eigenfunctions. But they provide you with a lot of additional information that you can maybe get from them in terms of, you know, which type of phytoplankton, what kind of CDOM, what's the slope, what's your particle size distribution. And so if you want more information on that, then you want to allow, maybe try to do a better job with the basis vectors. And so one of the things that I found would be really interesting is to try to get more information about the spectral variability. So my question to you, Oh, I already asked you that question. So then the next step was to take that model and actually expand on the number of phytoplankton. And so I grew a bunch of phytoplankton. I measured a bunch of phytoplankton curves. They all look like this, right? But they look like this differently because of all their pigments. And so using those, could I do a better job on the inversion? And so why should I limit to a single one when I have 6? So I took this experiment to South Africa. This is the Benguela upwelling current. I was working with my colleague, Grant Pitcher, who's a phytoplankton ecologist there. And the upwelling is what, the longshore winds drives the currents offshore. You get incredible nutrients up, and you get these expansive blooms that are these annual blooms. And what's really interesting about the system here is that the upwelling frequency is on the order of 3 days. I think it's on the order of more of 2 weeks on the west coast of California. So, is that right? It's like two weeks of winds. This is like 3-day pulses. And so you get this evolution of blooms, these really high concentrations that are just like every day you come out and the ocean's a different color. It's really incredibly crazy and beautiful. So I was on a ship, a wave washed over the porthole, it was green, and literally 3 hours later it looked like this. So there's tremendous spatial variability too. I had a little radiometer buoy, hyperspectral, and again, this is the 3-day difference in the watercolor. Yeah. This was not the good bloom. That was the one that caused diuretic shellfish poisoning, which was really fun for everybody on the trip. So I had a time series of measured reflectance. I used multiple phytoplankton eigenvectors, all of these in the model, and inverted to try to estimate the contributions. And then I validated with microscopy, because Grant looks under a microscope and he quantified the contribution of all of these different groups of phytoplankton. All right, so just to orient you, this is going to be date. We were out there for a couple weeks. This is spectrum from blue to red, and the brightness is the magnitude of the reflectance. So if, for example, I look at my first day, it's brightest here around 570. Right? Well, it's actually bright all the way through here. That's the bright part of the spectrum. And I've color-coded it. This was the color of the water. It was really turquoise and really pretty. And the blue is the measured and the black is the modeled. Okay, so here's what it looks like. And it's the pre-bloom condition. And what you can see is that, in fact, in this case, the measured and the modeled are right on top of each other. It did a really good job. And I'll show you the results of that. The next day, the water turned green. The model was a little bit not quite as good in the red. Then the water turned like really, really purpley red or orangey red. Actually, it was more purple. It looked more like this color. And it had these really strange peaks in the reflectance spectrum that were real. Then it turned orange, and that was the Dinophysis bloom that got everybody sick. And so then I have the estimates of each one of those phytoplankton contributions. And so this was the data that Grant Pitcher gave me about diatoms as a function of time, dinoflagellates as a function of time, dinophysis, mesodinium, and chlorophytes as a function of time. I took the cell counts and the size distribution and I converted it to absorption so that I could validate absorption against the model. Okay, so this is now a calculated absorption coefficient. And then I compared it to what the model output was. And the black is the model output of each of the different phytoplankton species. And so it's pretty tremendous because it was able to identify when I had diatoms and when I didn't, when I had dinoflagellates and when I didn't. The mesodinium, it was a slightly different magnitude, but it definitely identified 3 different blooms of mesodinium and chlorophytes and the bad dinophysis. But the thing that's also interesting is if you look at the Y scale, that's the magnitude of absorption. Diatoms and dinoflagellates were minor components. And yet, even when they're a factor of 10 lower than the dinophysis, you could still resolve the difference between diatoms and dinoflagellates when they're a minor contributor to the phytoplankton in terms of its optical properties. Because the spectral differences between them are so big, and the concentrations of phytoplankton in these waters was so high, that the features of absorption are really well detected. And so for a situation like this, when you have really high concentrations, you can do an excellent job of parsing out phytoplankton of different species using reflectance. It's pretty exciting. So the other thing, so these hyperspectral inversions, it's an overconstrained problem, right? Because we have, a couple of hundred measurements of reflectance. They're not independent. But it means that if we're only trying to estimate, 10 unknowns, we do a good job because it's overconstrained. And it allows you to kind of get at these gradients in reflectance and try and identify features associated with pigments. And that's why we do such a good job with the phytoplankton. And so if we can use this approach, then it allows us to start thinking about phytoplankton functional types, which Jeremy Wardell is going to talk about, thinking about pigment-based ones. And so I wanted to show you one more example from Allie Chase, who is a student here. Emmanuel, when was she in this class? Right. She just graduated from Bowdoin. She'd done an independent study with me, and then she was working in Boston but wanted to come back to science. And so this was part of her project? Yes, this is her master's, but I didn't know if she started it in the class. But she'll be here Monday. So she published a couple papers, and her master's was looking at the absorption spectra of phytoplankton, and this wasn't new. Other people had done this decomposition, but she took it into a really different realm. She decomposed the measured spectrum into the series of pigment Gaussians, right? And then she also added a little exponential for the non-algal particle, and this was a culture. But the point being that if then you can identify which Gaussian is associated with which pigment, or which series of Gaussians is, say, associated with chlorophyll, the harmonics, that then you could estimate by linear regression which pigments are contributing to your absorption spectrum. So that was great. It was a nice paper. Then she took it a step further and she said, well, why can't I just do the inversion instead of to a phytoplankton spectrum to the Gaussian spectra? And so she got a nice set of data where she had reflectance and pigments for validation. And so she ran the inversion. So she ran for every reflectance spectrum, she ran 8, Gaussians and CDOM and NAP. And her results were that of the 8 that she used, really 4 showed significance. And so this is total chlorophyll A, total chlorophyll B, chlorophyll C, and the photoprotective pigments. So this is measured by HPLC on this axis from a water sample, and this is retrieved by inversion. And so you see, it looks really good. I mean, that means that you can start to get pigment concentrations, right? Yeah.
Not quite clear. So she used 8 Gaussians for 8 different pigments, and it turned out that 4 of the pigments were
significantly related to the extracted HPLC. Yeah. So some of them are small that they just don't contribute. They're important diagnostic pigments, but they're not important absorption-wise. Okay. So the other part that I really liked about this paper is that she tested whether the inversion and the retrieval of the pigments gave her the significance of that retrieval. Was it any better than if she had just said, I have some global relationships between chlorophyll B and chlorophyll A, and if I estimate chlorophyll A, could I predict chlorophyll B just based upon pigment ratios? So she was testing whether the inversion model was actually getting you any more information on a global sense than just knowing the relationship between the pigments, right? And so she found that there were similar errors, but I guess I would argue that while the errors are similar, that the results are, I think, just much more intriguing to be able to think about where there are places where those pigment ratios don't hold. So in a global sense, there's a relationship between chlorophyll B and chlorophyll A. If you have more chlorophyll B, you probably have more chlorophyll A because you have more phytoplankton. But the actual ratio between those is going to vary as you move to sort of new ecosystem structure. And so while maybe for her data set it wasn't significantly better, her model allows you to look for change in ecosystems in the ocean. And that, to me, is a huge contribution. And I hope that when people read this paper, they don't just walk away with, well, like, oh, I just need to estimate chlorophyll and then use a pigment ratio and I can get whatever, because that's not the point of getting pigments, right? The point is to take it further. So she'll be here. You'll get to talk to her about it, but it's some really, really nice work. And so I'm going to end there. And any questions? Yeah.
I don't know if this is trivial, but could we go back?
We can definitely go back. Just keep telling me when to stop.
Yeah, here. Did you normalize the, because I see that.
Oh, yeah, these are normalized just so I could look at different spectral shapes.
Okay.
Yeah.
Because
There were huge differences in the magnitude.
All right.
Yeah, but I was truly trying to key in on, like, these were what was measured. But what went into the model was what I measured in the lab. So I took lab spectra for cultures and ran the model to try to estimate what I measured on filter pads in the field. And so I did not use these to run in the model. I used an independent lab data set. So again, there's still independence between what went into the model and what came out. Yeah. Mm-hmm.
Does it cause any problem if we say reflection is 1 minus absorption?
Reflection is 1 minus absorption. What do you, oh, in your model?
Like, yeah, not absorption coefficient, but like absorption, like capital A.
Absorbance. So if you're saying reflection equals 1 minus absorbance, you're back to incident light, reflected light, transmitted, right? And so you're making an equivalence here, and you're making a relationship there. So I think, yes, this is not the same as EU or LU over ED is proportional to backscattering over absorption. Those are different. This is basically doing a ray tracing.
So I'm asking because I've seen that in talks in like meetings, people use that because they have like spectrum of absorption. They want to get the calculate the reflection, and that's like they say that they calculate.
So reflection does not equal reflectance. Reflection is light comes in, light goes out, some of it's transmitted, right? Reflectance is the light that's emanating out of the ocean that contains the signal of what's in the water.
Right. Did I say
And this does not.
I meant.
Okay. Well, and I guess if you mean this, then, you know, basically what this is saying, is this absorption or is this Ken?
If you have a light filtered and you filtered out sample on top of it, the reflectance equal 1 minus the absorbance is sort of.
But that's in a spectrophotometer. It's not in the,
Some reflectometer,
yeah, but it's not, It's not for the ocean.
No, I mean, in particular, I'm asking about like corals that.
Oh, corals. So they're measuring the reflectance off of the coral. And so the difference is, and I had this discussion because one of the people on my committee was a person who looked at reflectance off of rocks to try to discern mineral content. And there's a difference between what's reflected off of an interface versus what ends up being in the upwelling light that's gone into the medium. And so I think that's what it depends on. Is this a filter? Is this a coral? Or are we talking about the ocean? And there's different physics going on because there's radiative transfer going on and there's absorption, scattering, and redirection, and part of that light comes in the upper field, but it's containing all this information. Whereas if it's an interface effect, it's very different. Yeah. And the loss here, presumably, by a coral is due to absorption, so that would hold. But it would not hold for the water. We have to go eat. So back here at 1:30, and we're going to, and there's some, Sasha, is the code on the site? Okay, just download everything. Cool.